Okay, so what we're doing today is histology. In histology, when you talk about histology, one of the first things you do is you break down the different types of tissue that there are. And there's going to be four that we're going to cover. We're going to cover the first one is going to be epithelia. And epithelia is important because about 90% of your cancers begin in epithelia. And then we're going to do connective tissue. And as we go along, this will get easier and easier because next is muscle, and there's just three types of muscle. And then when we get to nervous tissue, it will get easier still because there's really only 5 types of cells in your brain for the most part. Now this first whole drawing is just going to be epithelia, and then we'll switch the page and we'll do the next one. And epithelia is the toughest one. One, because there's several cell types, but also they arrange themselves in different ways. So I'm just going to break that down real quick, just in kind of a little bit of a connection or Venn diagram. I guess it's not really a Venn diagram, but you'll see where I'm going. Is you can have what's called a squamous cell, or some pronounce it squamous. And a squamous cell kind of looks like a fried egg. It's very, very flat. Or you can have a cuboidal cell. And these, as the name suggests, look like cubes. You can also have a columnar cell, which also, as the name suggests, look like columns. I'm just doing this in cartoon form for now, just so that we can lay out the different types. So those are the main different types of cell types. The next thing is that they can layer themselves in different ways. So you can have a simple layer. So if we did a simple squamous or simple squamous layer, it's 1 cell thick. So it's a layer of cells that are 1 cell thick. You can have stratified, which means there are multiple cells stacked on top of each other. And then you can have something called pseudo-stratified. It's called pseudo-stratified because when you look at it, it looks like it's stratified. But the closer you look, you realize there is basically just a cell layer thick. So when you look at it, it looks like there's multiple cells in thickness there, but when you come down to the basement membrane or the bottom, there is really only 1 cell layer thick, so it's called pseudo-stratified. Now the thing that gets confusing is you can have simple squamous cells, you can have simple cuboidal, and you can have simple columnar. Missed my line there a little bit. You can have stratified squamous cells, stratified cuboidal, and stratified columnar. The one exception is that there's really only pseudo-stratified columnar. So one of the ways to kind of organize this is I create just kind of a little bit of a Venn diagram, and I say, let's do squamous cells first. So these are squamous cells. And we can have these in a simple layer, or we can have this stratified. So it's a bit of a Venn diagram in the sense that what I'm going to do is talk about simple squamous cells and stratified squamous cells. You can do the same thing with cuboidal. You can have simple cuboidal. Maybe come back up and write this out. You can have simple cuboidal. And stratified cuboidal. And just in cartoon form, simple cuboidal would be a single layer of cuboidal cells. And those would be the nuclei. Stratified cuboidal you would have layers of cuboidal cells. Our last type of cell, that was cuboidal. Our last type of cell was columnar. And again, you can have those simple, or you can have those stratified. So you can have stratified columnar. Now the last remaining one is the pseudo-stratified, and since there's only pseudo-stratified columnar, I put that one down here. And again, it's kind of a little bit of a Venn diagram in the sense that I've got this combination, I've got this combination. I don't really want this to necessarily get in the way of these green lines. So that's why I'm making them really light, just showing you that there's a combination here and a combination here, combination here, combination here, here, and here. And the last one we have to do is there's also a combination here. So we have pseudo-stratified columnar. Pseudo-stratified columnar. Now, I will tell you that there's a couple of offshoots here. There's a couple of other things we need to get to clean this up. There's two different types of stratified squamous. There is keratinized. So keratin is a long stringy protein, and there's non-keratinized. And the way we'll end up drawing that, so we'll draw our stratified squamous. And we'll draw some fibers running through it. Maybe purple wasn't the best, but it'll show. So keratinized has fibers running through it. And non-keratinized, just as a heads up, they tend to be a little bit thicker of squamous cells because these line hollow organs like esophagus, and so they don't tend to dry out as much as our other example here, which is keratinized. Keratinized get very flat because they're external surfaces like your skin, so the cells dry out, which flattens them up a little bit, and that can help you identify keratinized versus non-keratinized. The stratified squamous and keratinized are very flat and very stacked together, and in fact the outer tissue is dead. And non-keratinized, this is going to be like esophagus, and they tend to be a little bit thicker of a stratified squamous cell. Another complexity here is we can have ciliated, pseudo-stratified columnar. And so we have cilia on it. And as you guess, that's going to line things like the trachea. And then we have non-ciliated. Pseudo-stratified columnar. So no cilia in this. And there's one other type of tissue that's kind of an offshoot. It doesn't fit into our whole Venn, and that's transitional. So we're going to just put that off to the side. And the characteristic of transitional is it's very stratified. But there's a lot of cells of different shape. So there might be really columnar looking cells, and there might be really flat and cuboidal cells or squamous cells. There's just a lot of cell shapes in the stratification. And the place you're going to find this, we'll come back to this, is when you're moving from one type of epithelia to another. So if you're going from, say, a simple columnar up to a simple cuboidal, up to a simple squamous cell, and this is what happens when you go down the airway, and that kind of makes sense, is at the top of the airway you want a thick cell so that you're protected from the bacteria that you breathe in. But by the time you're deep down into the lungs, you want air to pass through those cells. You want air in the alveoli, the air sacs, to get into a capillary, so you'll need a nice thin barrier. So as you go down, you have to transition from one type of cell to the other, and so you use a transitional epithelia. The other place you use a transitional epithelia is in the bladder. So the two places you use transitional is transitioning between types of epithelia. And the other classic example is the bladder. So this is an example of transitional. So all of this around here, the whole layer, all of these cells in here are transitional epithelia. We see it over here. The whole thing is transitional epithelia. And you get the idea that this is, if I told you that this is bladder, that would all make perfect sense, because you get the idea that you could fill this up and stretch this out, and then you would still have a continuous layer of cells. And so transitional epithelia means that you have lots and lots of different cells. And if this were to stretch, if you were to fill this with fluid so that this would have to expand out, or this would have to expand out, you would still have plenty of cells to make a barrier. Now here's where I think is the real key to understanding histology, is you bring in the principle of complementarity. Can you look at a tissue type and kind of understand what it needs to do? And I think this is why I kind of like this method other than other like sorting methods, is because when I look at a simple squamous cell or a simple squamous cell, it tells me that I want one of two things. Either, I might switch colors here just so it's a little bit clearer. Either I want something to easily pass, and in that case what I want is I want permeability. So I want things to go through. And the example there is air sacs of the lungs. And I'll show you a picture of this. Okay, so what we're looking at here is, it's kind of like we're looking down into the lungs. And this area right here is called a bronchiole. So this is way down deep in the lungs. And these are kind of little air sacs, and they're called alveoli. And when we cut through them, we see something like we see down here. What we're seeing is perhaps this is a simple squamous cell. Perhaps this is a simple squamous cell. Perhaps this is a simple squamous cell. It's pretty hard to tell exactly what a simple squamous cell is, because you cut through here, you may not see the nucleus and things like that. Another good example of where you're going to see simple squamous cells are if you have a blood vessel. That wall of that blood vessel is going to be lined with simple squamous cells, and the reason is it allows blood to flow smoothly over that smooth surface. Another example is if this is the heart. I'm not much of an artist. And you have 4 chambers. The chambers of the heart, or the inner layer, is lined with something called endocardium. And that endocardium is really slick because it's made of simple squamous cells. The other thing I may really want is I want a smooth surface for something to flow along. And that's why you use this type of tissue, simple squamous, on the inside of blood vessels. Line blood vessels. Another example of that would be the endometrium of the heart. Now, why you might want cuboidal is because maybe you need some structure, or that cell needs to make something. The classic example, and where you're going to see this mostly, is in tubules or ducts. The main classic example that we'll see in lab is kidney tubules. So you want something with a little bit of a structure so that you can make a tube. A simple squamous cell would not make much of a tube. So what we're seeing here is kidney tubules, and you have to be a little careful when you're locating these on the slide because you're going to be looking at a slide of the entire kidney, and so you're going to see some other areas where you're not seeing simple cuboidal. The best place to look is where you see these cross-sectional tubes, and what we're seeing here is a cuboidal cell with its nucleus, another cuboidal cell with its nucleus, and so you can kind of follow all the way around, and you can see that this is a simple layer of cuboidal cells, and so this is simple cuboidal. What the kidney does is it filters out a lot of the fluid out of your blood, but you don't necessarily want to urinate all that out, so you have to have all these tubules that fluid goes through, and when it flows through there, you get to pick out the stuff that you want to keep. So you want to take back the water and the sodium and the sugar and things like that. So when you filter out all this water, you have to have all of these tubules in order to get this stuff back into you. And that's why you need kind of a nice structure on a lot of tubes, and that's why you use simple cuboidal. What you want with columnar is either that cell has to make a lot of stuff, or you want a lot of protection. And by protection, I mean maybe it's lining your digestive system, and this is a little bacteria. You don't want that bacteria to be able to get between these cells. So having a nice tall cell is more protective. It allows you to keep bacteria on the outside of something rather than letting it through. And so the main example of a simple columnar is it lines the digestive tract. So they'll look kind of like that along the whole, and there'll be a whole lot of other tissue around it, but these line digestive tract. And if you look at digestive tract columnar cells, you'll realize that they have a lot of protein making machinery so that they can take up nutrients, and also immune elements so that they can protect the inside of the gut, protect bacteria from getting through from inside the gut into the rest of the body. And so there's a lot of immune and protective elements in there too. So you need a nice thick cell in that case. This can be an example of where it's kind of difficult to do histology because you don't know exactly where you're looking at all times. You don't really know what you're looking for. What we're looking for here are these cells right here. So there's a nice thick nucleus in there, and you get the idea that these are very nice columnar cells. We also see them over here. And what we're really looking at is in your digestive system, you have these things called villi. And if I wanted to be more accurate, I'd say on top of those are microvilli. And this increases the surface area for you to be able to absorb nutrients from your digestive system. So if I can focus on this right here, it would look kind of like this. And it would be lined with simple columnar cells. And what we're looking at specifically is kind of a section right through here. So this is this, and this is over here. But you get columnar cells, simple columnar cells on both sides. When I look at stratified squamous, the main thing that I see is you could take and cut a huge section out of this, and you still have cells protecting. So the main purpose of stratified squamous is that you can slough cells, so cells can fall off, or you can simply lose cells by abrasion. And so the example here will be skin. And again, so you can scratch your finger across your skin and you can scrape off a bunch of cells and you'll still have a barrier. The same thing is in non-keratinized, but in non-keratinized, without the keratin, it's not quite as strong. So the keratin makes this stratified squamous really, really strong. So your skin is pretty durable. It's pretty strong. You have other places like your esophagus where you still want to be able to slough off cells, but maybe it doesn't have to be quite as tough as skin. So when you eat food, it slides down your esophagus and it can scrape off some cells. So you definitely still want this ability to scrape off cells and maintain a barrier, but it doesn't have to be quite as tough as keratinized. The other difference, since this is an internal structure and it stays moist, whereas the skin is an external and it dries out, usually a non-keratinized stratified squamous will look thicker. So these are not necessarily really the best slides, and I apologize for that, but that's just kind of what I have accessible to me today. It's like, for example, this kind of hollow space is just kind of a fracture in the slide, and it's not necessarily normal. What we're looking for are, it can be difficult to imagine, but there are these tiny little flat cells, and we extend out way up here. So we've got this layer after layer after layer after layer of squamous cells in here. You can kind of see the cells start down here, and this is where actually the mitosis occurs. So these are where the cells are being made down here. And you can kind of get this idea that they do look like eggs, and so they are squamous cells. By the time they rise to the surface of the skin, they flatten out and they dry out, and they're dead from here on out. But it's definitely a stratified squamous layer. You see multiple layers of cells. You can't really see the fibers, but you can get the idea that the fibers are in there in some places where it looks kind of wispy in here. Now when it comes to non-keratinized, again, you see that same kind of squamous cell. And again, the area of active mitosis is down here. And so these cells are growing out in this direction, and as they grow out, they flatten out. But again, because this is a moist surface, this is the inside of the esophagus, it doesn't necessarily dry out necessarily as much as skin, and so you don't see the really super flat cells that you see in skin. So there's keratinized with really a lot of flat cells, very flat because they're dried out. And this is non-keratinized. Now stratified cuboidal, in all honesty, is pretty rare. So this one is rare in the body. So we're just going to write that this guy is rare. But if you're forced to give an example, you would say sweat glands. Stratified columnar, it's also really rare. But if you're forced to give an example, you'd say the pharynx. And the reason for that is a stratified columnar is pretty durable. We said over here that simple columnar is very protective. Right at the throat, you might want a double layer of cells, something really thick to make sure, to really make sure that if there was a bacteria on here, it can't penetrate, and the throat would be one of the best places to have that kind of layer. So that's why it's in the pharynx. Ciliated pseudo-stratified columnar. This is in the trachea. And again, that's a good place to have a nice thick columnar cell for protection. But you add the added need for cilia. You have cilia down in your lungs and in your airways that will sweep bacteria up towards your esophagus and digestive system. So I've said this a couple of times, that it's hard to orient yourself when you're looking at histology, and that's probably especially true on this structure. This is the trachea. One of the first things you're going to need to do is you're going to need to look for the cilia, and it's very hard to see, but you're going to have to focus up and down, and you see these tiny little wisps in here. And that's the cilia. It almost takes more imagination than anything to see them. But here, right here, is a pretty good spot where you can see. You get this idea that there's a cloud of something there. There's a little wisp of cilia. And so this is also pseudo-stratified because if you looked along here, almost every cell touches the line. So it really is a single layer of cells. But it obviously looks very thick. It looks like there's multiple cells there. So that's why it's pseudo-stratified. So again, maybe this is a cell, and this is a cell, and this is a cell. But all of them seem to come back and touch the basement membrane. So that's why it's pseudo-stratified. I don't have a slide of non-ciliated pseudo-stratified, but you would find those essentially in sperm ducts. And it would look very much the same. It just wouldn't have that cilia. Non-ciliated pseudo-stratified columnar. It's relatively rare, and it's one that we won't actually draw in lab, but it's in sperm ducts. So I think by lining up the type of cell with the three choices of it can be simple, stratified, or pseudo-stratified, you kind of see that there's an organization to the epithelia that makes it a lot easier. And then I think one of the keys to memorizing or being able to identify a tissue when you look at it is being able to imagine kind of what it's trying to do. So in lab and on lecture, you'll be asked to identify tissue in a microscope, or on lecture, you'll be asked to draw the different types of epithelia and then give an example tissue of where you might find it. And the best way to answer that question is to really look at the tissue and figure out what it might need. Next, we'll move on to connective tissue. So again, we're doing histology. And in histology, we're talking about four main types of tissue. We're talking about epithelia, connective tissue, muscle, and nervous tissue. And in this video, we'll do connective tissue. Connective tissue is not nearly as complex as epithelia, but it's still a lot more complex than muscle and nervous tissue. The first thing is you have something called connective tissue proper. So connective tissue proper. And that can be divided into two different types. You can have loose, or you can have dense. You also, within this, you have cartilage, bone. And I'm spreading these out a little bit, you'll see, because there's different types of cartilage. And there's different types of loose and dense. The different types of loose connective tissue proper are areolar, adipose, and reticular. The different types of dense are regular and irregular. The different types of cartilage are hyaline, elastic, and fibro. Bone is pretty much just bone, and blood is pretty easy to recognize because it's the only liquid tissue in the body. So these are the main types of connective tissue. We've got connective tissue proper, that's loose, that's areolar. And where we usually find this, it's kind of a loose tissue, and what it does is it wraps organs. So you can find this as an external covering to internal organs. So this is loose areolar, and you get this idea that there are cells in here, but you also see lots and lots of fibers in here. And those fibers basically create a covering for certain internal organs. So this is loose areolar. Adipose is fat storage. Now, just looking at this, you might notice you have to be a little bit careful in distinguishing adipose from simple squamous, because it kind of looks like lung, it kind of looks like this is full of air, but you see more regularity here, and you don't see bronchioles and things like that in here. The other thing that you'll notice is when you catch the nucleus, it kind of looks like, sometimes they look like rings, because we've got a cell here, and here's the nucleus, and so that's a adipose cell, or maybe there's another one here. And the nucleus could be this guy right here, or
they have a nucleus off to the side and they're full of fat. So they look kind of like rings with a pearl on it or something like that.
In reticular, where you're mainly going to see that is in the spleen and the lymph nodes, and it's kind of an internal skeleton for certain organs. So it's skeleton for organs.
So I don't have a slide from class of reticular. This is actually a slide from Wikipedia, and it's staining reticular tissue in the liver. And what you'll notice, there's these long fibers, just all kinds of fibers. And we'll see this, especially when we do lymph nodes, and maybe the spleen too. The reticular fibers are like an internal skeleton for certain structures like liver, spleen, and lymph nodes.
Dense regular, when you look at it, it looks very in line. So the tissues look very linear. And I think if I asked you to think about something in your body that's very long and straight, you'd probably think of tendons and ligaments, so long and strong. Irregular, when you look at it really close, and I'll show some slides, it looks like the fibers are kind of going in all directions. And the way that you see this is when you want kind of large coverings, like over joint capsules. So there's coverings over your joints. And then also you'll see this under the skin, so the dermis.
One of the things to be cautious about is this sometimes can be mistaken for skeletal muscle. Skeletal muscle looks very like long and cylindrical. The way that you know that this is not skeletal muscle is there are no striations. In a skeletal muscle, you'll see these kind of striations, these kind of stripes. This tissue does look very regular, though, and so this is dense regular. And if I asked you, is there a connective tissue in your body where you'd want it to be kind of long and linear and strong, you'd probably say ligaments and tendons. The idea behind dense regular is they need to be strong in this direction. Now, I don't have a slide of dense irregular, but imagine if rather than being all lined up like this, everything was kind of mixed up. All the fibers were going in different directions. And then you would have dense irregular. There'll be a slide in lab. I just don't have a picture of it now. So whereas when they line up like this, that's tendons and ligaments. When they line up like this, it's like you might be pulled in any one direction. And so this is the dermis. This is the layer in your skin, the lower layer in your skin. Or also, if you have a covering of connective tissue over a joint, then that would also be dense irregular.
When I think about hyaline cartilage, I always think the cells just kind of always look the same. They kind of have these big pink nuclei. And the difference between hyaline, elastic, and fibro is the cells always kind of look the same. It's just in hyaline there's no fibers. This is your good old run-of-the-mill cartilage, like nose. Costal cartilage, like your ribs. And then joints. If you were to take these chondrocytes, these cartilage cells, and put in these deep kind of purple fibers, these are elastic fibers. That's how you can spot. They're even probably darker purple than that when they're stained. They're really dark. And that makes the cartilage really, really stretchy. And so examples of this are your ear, and then something called the epiglottis, which is the little flap that helps separate food as it goes down your throat. It covers over your airway when you swallow food, so that food goes down your esophagus and not down your airway. When I look at fibrocartilage, they have these really kind of wispy blue fibers running through. And these are collagen fibers. And collagen is super tough. It's like the body's duct tape. It's like if you wanted to take a little hunk of your skin, pinch some skin, and try and pull it off right now, it's the collagen fibers in there that are being really strong so that you can't pull anything off. The main examples of this would be intervertebral discs. So you imagine if you wanted a really strong cartilage somewhere, Where would you put that? You'd need to put that between the vertebrae or the meniscus of the knee. Another super stressed piece of cartilage is the cartilage that's between your bones of your knee. So between the tibia and the femur.
This is cartilage, and one of the things that I always identify cartilage with is these chondrocytes. These chondrocytes, they have these beautiful purple nuclei. And the thing about cartilage is you'll always notice these chondrocytes. They look the same whether you're in hyaline cartilage, fibrocartilage, or elastic cartilage. The way you know that this is hyaline cartilage is you don't see a bunch of fibers running through it. That's a little hard to distinguish what you don't see. So we'll move on to elastic cartilage next. So here again, this is cartilage, and you can kind of see that nice chondrocyte, cartilage cell. And again, it has that nice purple nucleus. What we're seeing in this slide is tiny, tiny little fibers. And by tiny, I mean they're very distinct and they're thin. And these are elastic fibers. In your body, you have two main types of fibers. You have a third that's reticular, but the two main types are elastic, which are really, really stretchy. So when you pull on your skin pops back. That's elastic. You also have a really strong fiber in collagen. And I consider collagen kind of the body's duct tape. It's really, really strong. We'll see that in fibrocartilage. Now, it's a little hard to see this, and honestly, in different slides, you're going to see it stained different colors, but you get this idea that there's huge fibers running through here. It just looks like there's stuff in there, and that's the collagen. So if I flip back really quick, you don't see that kind of pink fiber in hyaline. And it's not a thin fiber like an elastic. It's these huge, big fibers. So this is fibrocartilage.
Now bone, I'm not a particular artist, which is why I'm going to mainly just rely on going to show you some slides. But bone is usually pretty easy to identify because there's these concentric rings with the actual cells of the bone being interspersed through. So that's bone.
This is bone. Bone is pretty simple because you can see these really concentric rings. And then the little bone cells. You have different types of bone cells, so we'll keep it simple at this level. But you see these concentric rings and the little bone cells.
And blood is fairly simple because you'll see tiny little blood cells. And then occasionally, when you're looking at a smear of blood, you'll see these little purple cells. We'll talk about those, the different types of cells. I'm trying to draw a lymphocyte there, a monocyte there, and I was trying to draw neutrophil there.
This is blood. Obviously, this is pretty simple. This is the specific blood cell. Unfortunately, I don't really see any in here. I don't really see any white blood cells. You get the idea that there's one down here, but you can't really see it. This little purple guy and this little purple guy. So when you look at blood smears, purple means it's a white blood cell, and all the other cells are red. So once in a while, you'll see tiny little specks. I don't see them on here, but that would be platelets. Maybe right here are platelets.
That's connective tissue. So again, we're doing histology. And sorry to be so repetitive, but sometimes it's just good to hear things multiple times so you get them in. The four main types of tissue. When you talk about histology, is epithelia, connective tissue, muscle, and nervous tissue. We're going to do muscle in this one. And muscle's pretty easy because there's really only three types of muscle, essentially. You have skeletal, cardiac, and smooth. When you look at skeletal muscle, it generally looks very long. And usually, if you're looking at a slide, you're not even seeing joints between cells. You're just seeing very long pink columns. And you can kind of see some striations, some stripes. I'll show you real pictures, but I just want to get something on the paper. And we'll talk about these stripes when we get into muscle physiology. But basically within this area, if I were to focus on this, within this area, there are fibers called myosin and fibers called actin. And when muscle contracts, these fibers slide against each other. And so when you look at muscle, deep down in there, you have these little layers. So this layer right here ends up looking kind of reddish. I guess this whole layer here looks kind of reddish. And then this layer here does not look so red. And so you end up seeing striations. Some other things are the nuclei. There's multiple nuclei in these cells. And they're off to the side. When we look at cardiac muscle is branched. And so, whereas skeletal muscle, everything looks pretty tightly packed together. There's much more space in cardiac muscle. You can see more airspace. You see those same striations? And another thing you see when you see a joint between two cardiac cells is you see a really deep purple area. And this is called an intercalated disc, or can also be pronounced intercalated disc. So that's cardiac muscle. Smooth muscle, usually when you look at it, it's also very tightly packed. But if you look very close at the cells, the structure is called a spindle-shaped. They look like tiny leeches, long worms. That's what it looks like. So what we have here then is when we look at skeletal, cardiac, and smooth, the way that you can tell them apart is you base it on 1, the shape, 2, nucleus location, 3, presence or absence of striations, and 4, presence or absence of intercalated discs. So in skeletal, they're described as cylindrical. In cardiac, they're kind of branched. And smooth, they're called spindle-shaped. And that just simply means they look kind of like that. In skeletal, there's multiple offset, so to the side, nuclei. And the reason that is skeletal cells are born as separate cells, or they develop as separate cells, but then they fuse together. So you'll see multiple nuclei in a fiber of skeletal muscle. Cardiac, honestly, I see it both ways, but I generally describe them as there's a single nucleus, and it's offset. Sometimes people describe it being as more centered, but I tend to call the nucleus off to the side. So it's off to the side. And then in smooth, there's a single centered nucleus. So the nucleus is right in the middle. Skeletal muscle has striations, so we'll just write yes. Cardiac muscle also has striations, so we'll call that yes. And again, that's those actin and myosin filaments causing that coloration. And smooth, there's no striations. There's still actin and myosin, but it doesn't line up so nicely, and so you don't see the striations. Intercalated discs, again, are special to cardiac cells, and what it does is in a cardiac cell, there's no nerves running along the muscle fiber to stimulate each muscle fiber like there are in skeletal. So if you have an electrical current, and we haven't really learned about this yet, but you have an electrical pulse that travels from one cell to the next, the intercalated disc is really just a bunch of gap junctions that allow that electrical pulse, what it really is sodium and calcium ions, to flow from one cell to the next cell. And so cardiac cells do have intercalated discs. but skeletal does not, and smooth muscle does not.
This is not our best slide of skeletal muscle. Usually what skeletal muscle looks like is just really long bands, and you see these striations, and the nuclei are off to the side. The problem is in this particular slide, this particular picture, it was cut in this direction. So you're seeing cross sections of those fibers. Where the actual striations come from is when you look deep down into the muscle, and we'll get to this when we talk about muscle, is let's say that we can expand this out. In that stripe, you'll have myosin. And in between that myosin is actin. And you get the idea that when you look right here, you kind of see a red stripe. And when you look right here, it doesn't look quite as red. And so the striations, and you can kind of see them right up here. The red stripe, kind of the red stripe, is where you see the myosin. And the white stripe is where you don't see the myosin. So that's skeletal muscle. This is cardiac muscle, and one of the first things you'll notice is it just looks like there's lots of space in there. And the reason there's space is because these cells branch. Now don't actually see the nucleus in there, but you get the idea that these cells will split and branch. Maybe there's another one here. Kind of have to be a little bit imaginative. The other structure that you can note really clearly is this right here, that kind of line, or I'll point to one rather than mark over it, or this one right here. That's called an intercalated disc. In cardiac muscle, if I can draw this larger, you need electrical current to go through the cell, and it needs to split and go in multiple directions. But what actually is happening in that electrical current is sodium and calcium are moving from one cell to the next. So this structure right here is really a place where there's lots of little holes where sodium and calcium can go through, and that's called an intercalated disc. Or sometimes it's pronounced intercalated disc. This is smooth muscle, and as with a lot of these slides, you have to be somewhat imaginative that these cells are kind of spindle-shaped with the nucleus in the center. So you don't see any striations, but you do see centrally located nuclei. So if I can flip through those quick, there's no striations here, no intercalated disc. It's a spindle shape, and the nucleus is in the center. When I get to cardiac, I see intercalated discs. I do see striations. They're hard to see in this slide, but I do see striations. I see a branch structure, and the nuclei are typically off to the side of the cell. Skeletal, if it was a better slide, I would see nuclei off to the side. I would definitely see striations, and I see large, long cylinders of muscle fiber.
So the last time we're doing histology. And histology is the study of different tissue types. And the four main types of tissue are epithelia, connective tissue, muscle, and nervous tissue. And this time we'll do nervous tissue. Nervous tissue is the simplest of them all because really there's only 5 to 6 cell types. There's 6 cell types, but it depends on if you're talking about the central nervous system or the peripheral nervous system. The main cell type is a neuron. And a neuron is a little bit difficult to draw. I always draw them kind of like this. We have a nucleus. This is called the axon. Usually the neuron contacts other neurons here at what's called the synapse. This is the soma. These guys out here extend out as well. And these are called dendrites. So that's the main excitable cell of the brain that basically makes decisions for you. We have another type of cell over here, which is a support cell, which is called glia. And in the central nervous system, let's just do it that way. we have an astrocyte. What it does is it kind of looks like it's a huge star-like cell. That's what astrocyte means. And what it can actually do is say that we have a neuron running over here. And its axon is running like here. The astrocyte can actually communicate with the neuron and communicate with perhaps a capillary over here. So a blood vessel. And the astrocyte will take things out of the blood and deliver it to the neuron. And we call that the blood-brain barrier because the astrocyte determines what comes out of the blood and what makes it to the neuron. So that's what an astrocyte does. We also have something called a microglia, which kind of looks a lot smaller than an astrocyte. Maybe not a lot smaller, but it kind of looks kind of spiderish. We won't necessarily be drawing these, but what this does is it's an immune cell. We have something called an oligodendrocyte. And an oligodendrocyte, if we have a neuron going through here, an oligodendrocyte is a cell that will kind of extend feet and wrap around a neuron. So maybe another one goes over here and another one comes, and it will extend and it will wrap around a neuron multiple times. And that provides the myelin. So it does something called myelination. And if I can skip over to peripheral nervous system, you have a similar type of cell in the peripheral nervous system, so outside of the brain and the spinal cord, that's called a Schwann cell. That does the same thing, only in this case it's kind of a flat cell with a nucleus out here, and then the neuron would be going through the middle. So it kind of looks like a fried egg that's just wrapped around an axon. Our last CNS nervous tissue cell is called an ependymal cell. And these look a lot like epithelia. And this would be inside of a ventricle. So inside of your brain you have open spaces that are filled with fluid, and they're called ventricles. And then this would be neurons and things like that would be up here. So there would be fluid in here. And ependymal cells line those ventricles. So it's much like an epithelia. Kind of lines the digestive system and lines the reproductive system and the respiratory system. Ependymal cells line the ventricles in the brain. Alright, so that's all the nervous tissue. The only two that we will draw is we will draw this one, and we can draw this one. Because usually you'll see them in the same kind of slide. We'll look at something called ox motor neuron, and this will be the neuron, and you'll see a lot of tiny little purple cells around it, and those are the astrocytes. The last one is neuron, and this guy right here is my neuron. It can be hard to tell which part of the neuron is actually the axon, but let's call that the axon. This is the soma. These out here, these guys out here are the dendrites. And then the synapse would be well out here. So this is the neuron. The only other cell that we can really identify here are these little blue guys, are astrocytes. So that's nervous tissue.